Well, let's take a moment and review over the urinary system. On this particular presentation, we will be reviewing over the parts of a sheep kidney and then the histology of the urinary system. This is a typical sheep kidney, however, a cow kidney would look very similar. Now, as we look at the kidney, let's just point out a few key components. Right here on this outer edge, if we were to peel a very thin layer away, and we're talking about the thickness of the skin of an onion, we would have a capsule, and it just covers and holds that kidney in place. But with the kidney itself, it is divided into two regions. This region right here, which is our renal cortex, and then this area right here, which is our renal medulla. So the two main divisions of the kidney are the cortex and the medulla. There are structures associated with each area. Now notice, and we're going to draw this out to make it even clearer, look right there at that structure. Notice that that is a triangle. That is our renal pyramid. So we'll take that off. But if we were to take our pointer here and look at it, look at that right there. Look at it right there and there. Those are the renal pyramids. As we move to our future slides and we start looking at our proximal convoluted tubules and our distal convoluted tubules and our collecting ducts and all those components, we're going to find that they're located right within that region, and that there are millions of these structures found within each of our kidneys. Running in between the renal pyramids, we have renal columns, and we have those labeled right in here. The tip of the pyramid is called the renal papilla, and so we have the renal papillas labeled on some of those areas. This area right here is the renal pelvis, and then we have the ureter, which will leave the kidney and it will continue down and reach to the bladder. Here we have a second view of the kidneys. Again, we'll have the capsule here. We have the cortex clearly labeled. Notice the color change between the cortex and the medulla. Again, you'll notice that we can see our renal pyramids, the tips being the renal papilla. The area right in between is going to be the renal columns. We have the renal pelvis, and then right there we would have our ureter. So what is within these medullary pyramids? Well, the first thing I hope that you recognize is that we have seen this slide before. And these structures right here that we're looking at, early on when we were studying histology, we used these slides and these structures to learn to identify simple cuboidal epithelium. So you are seeing simple cuboidal epithelium throughout this slide, as those types of cells are very prominent. But in addition to that, we have this very large structure there and right there. And that represents Bowman's capsule, or the glomerular capsule. Now, we've talked about this in lecture, but we do have more blood entering the capsule than leaving, and we have a bottleneck of blood within here, and we have the separation of the blood cells from the plasma so that we can start to separate the waste from the blood. And so we have our glomerular or Bowman's capsule. Now, these simple cuboidal epithelium structures are the convoluted tubules, and we'll look at how you differentiate the types of these tubules on the next couple of slides. Here we have the glomerular capsule, or Bowman's capsule again. And again, when we're seeing here, we call it Bowman's capsule because Bowman was the individual who identified this structure. So now as we begin to look at our convoluted tubules, how does one differentiate a proximal convoluted tubule from a distal convoluted tubule? Well, first of all, we're going to be looking at these structures right here, these areas of simple cuboidal epithelium. Now, leaving Bowman's capsule, the first convoluted tubule that is reached is the proximal convoluted tubule. Now, we're going to tell the proximal from the distal by looking at this open area or lumen. Now, because the proximal convoluted tubule is reabsorbing any waste or material that can be reused by the body, we're going to have an increased surface area. And that's going to be demonstrated by this fuzzy appearance. So anytime you look and you see a fuzzy appearance, that's going to indicate a proximal convoluted tubule. And as we continue through the nephron system, we'll go from the proximal convoluted tubule to the loop of Henle, and we'll have our descending and our ascending loop, and finally we will reach our distal convoluted tubule. Our distal convoluted tubule is no longer absorbing material, it's transferring waste out of the kidney. And so that's going to be indicated by a clear lumen. So the way we're going to look at these is that both of these are going to be represented by simple cuboidal epithelium, but to differentiate the proximal convoluted tubule from the distal convoluted tubule, we will be looking at the lumen or the hole of the donut. If we have a clear, then we're going to have a distal convoluted tubule, where if we have a fuzzy lumen, we will have a proximal convoluted tubule. Finally, the last tubule system that is encountered is the collecting tubes. Now, the way we tell the collecting tubes is not by the lumen or the hole of the donut, but it's by the overall appearance. And while we use the exact same stain to stain the entire slide, the proximal convoluted tubules and distal tubules will appear a darker pink. But our collecting tube, which is what we see all through this slide, appears a much lighter pink. So look for the light pink, we'll call them donuts, but structures, and that's how you will identify the collecting tubules.